Hi, everybody. Welcome to this week's episode of the SBGrid webinar series. Hope everything's going well for everybody in their various stages of reopening or lockdown or everything else that's going on in the world. Hope everybody's staying safe. I have the great pleasure to introduce Steve Reichow from Portland State University. Steve did his PhD at University of Washington in Gabriele Verani's lab, and then postdoc at University of Washington and HHMI, and now started his own lab at Portland State University. And today he's going to talk to us about the Connexin-46 50 gap junction channels and dynamic lipid environment resolved by Cryo-EM. So with that, I will turn it over to you, Steve. And thanks again for presenting today.
Yeah, great. Thanks, Peter. Let's get the screen pulled up here.
All right, looks good.
All right, wonderful. Yeah, thanks again for the invitation and giving us this kind of venue to share our work. And to all of you who are in the audience, I can't see you, but looking forward to telling you about yeah, some exciting work that's been coming out of our lab here at Portland State. So this graphic I'm showing you here is really kind of meant to represent the two major research focuses in my lab. The first is on vision biology, and in particular, my lab is interested in the eye lens and the molecular mechanisms that is used by this organ to maintain transparency throughout our lifetime to support vision. And at the center here is one of the very important membrane channels to maintenance of lens transparency, a gap junction. And so another focus of our lab is, in general, trying to study and understand these pore-forming membrane channels. And so, yeah, once I've started my lab here at Portland State, a major focus has been on trying to use Cryo-EM to visualize these cell-to-cell communication channels that are abundant in the eye lens and throughout our body. And I'll be telling you a bit about that work today. Let's get a laser pointer here. Yeah, so if you're not familiar, a little background on the gap junctions. So these are ubiquitously expressed membrane proteins throughout our body. And I refer to them as cell-to-cell communication channels because, well, that's their physiological function, and they fulfill that function through a unique molecular architecture. So that's meant to be illustrated with this diagram here. And one of the first unique feature of these channels that you'll notice is that they actually transpose through two membrane bilayers. So they form a continuous channel that connects the cytoplasms of two adjoining cells. The other unique feature of these channels is they have an unusually large pore, right? So the pore of these channels is roughly 1.5 nanometers in diameter, and that's sufficient to permeate many different molecular substrates. So not only things like water and ions, but larger molecules such as nutrients, cell signaling molecules, hormones can permeate through these channels. And in this way, they facilitate both short-range and long-range communication between cells and tissues throughout our body. So you may be most familiar with these channels and their role in facilitating electrical coupling in organs like the heart and the brain, where they form what are known as electrical synapses. So in the heart, these channels play a fundamental role in allowing your cardiac cells to synchronize the heartbeat that you feel throughout in your body. And in the brain, they also have a role in electrical synapse formation. And in this movie, what I'm showing here is actually some long-range calcium signaling mediated via the gap junctions in a glial cell network. But these channels aren't just expressed in electrically active tissue, they're expressed throughout our body and play an important role in maintaining homeostasis, facilitating development and long-range communication. And in our lab, we use the eye lens as a model system to understand how these channels work. And so in the lens, they play an important role in establishing what is known as a microcirculatory system of the lens. So the lens is an avascular organ, and in order to deliver nutrients and remove waste products, it's evolved this highly coupled system of membrane channels and transporters that bring nutrients deep into the lens tissue and then recirculate waste products back outward through these gap junction coupling channels. And so there's a wide diversity of physiological roles that these channels facilitate, and this is performed by various types of isoforms. So in the humans, we have 21 different isoforms that are constituted by proteins known as connexins. And so each of the various connexin isoforms have their cell type specific expression and unique channel properties. So when I say that, I mean distinctive properties in substrate selectivity and gating and other forms of regulation. And to add to this complexity, these various isoforms, many of your cells will express more than one isoform, right? So, and many of these isoforms can co-assemble in complex ways that we don't fully understand. This is another kind of ongoing question in the field and in my lab, is trying to understand the mechanism and functional significance of this kind of heteromeric assembly of these channels. Now, because of these kind of fundamental roles in biology and physiology, aberrant function of the gap junctions is associated with a variety of human diseases. These range from inherited deafness, blindness, arrhythmia, stroke, and some cancers. And so because of these fundamental roles, these channels have been of interest to for many decades, not only to cell biologists, but also to structural biologists. In fact, the gap junctions are perhaps some of the longest studied channels in the field of electron microscopy. In fact, it was visualization of these channels in cell sections that earned these proteins their name. And so here's an example of such of an image. This is a cell section from a crayfish neuron. So this is an electrical synapse at these neurons. And what you're looking at here is actually a large array of gap junction channels that are coupling these two opposing cells. And so these striations here are the plasma membrane, and these puncta that are arrayed across these membranes are the gap junction channels. And so at a morphological level, these structures are identified by a characteristic 3.5 nanometer gap that is imposed by these channels that couple these two membranes to one another. And so attempts to reconstitute these types of gap junctional arrays in an in vitro type system have demonstrated that both the assembly and channel function of the connexins is dependent upon the particular lipid environment. And this is a phenomenon that my lab has become very interested in trying to understand. And in particular, this marries with our interest in the eye lens. So the eye lens has a lot of unique cellular adaptations, and one of those unique aspects is the lipid environment within the lens is constantly changing, and it changes with age. And one of the things we're interested in trying to understand is if those age-related changes in the lipid environment may contribute to the common disease of the eye lens that occurs as we get older, known as cataracts, or loss of vision because of the degradation of this organ. And so in order to study these channels in a lipid environment, well, let me step back. Because of this kind of highly ordered state that these channels natively assemble in the plasma membrane, they've been a target for structural biologists using methods known as electron crystallography. So reconstituting these channels into a lipid bilayer to study by diffraction methods in an electron microscope. And so if I were to turn this plaque and face, you would observe these channels in a large two-dimensional array. And so by reconstituting these arrays in vitro, we've been able to study their structure using electron crystallographic methods. And here's kind of a snapshot in time over the progress that has been made by studying these channels using this kind of method. And so these studies go all the way back to the 1970s by Casper and colleagues, reconstituting the liver gap junctions into these, beautiful hexagonal arrays. And you can see as the technology progressed over time, more and more structural detail was able to be coaxed out of these systems. And so some of the first structural details that were elucidated was that these channels are formed by an assembly of 6 monomers in what is known as a hemichannel. So we're looking down the pore of the channel. The pore is very large, again, making sense with its function to facilitate the transfer of a wide variety of molecules. In the late 90s, Vincent Unger developed the first 3-dimensional reconstruction of these channels at around 8 angstrom resolution. And so at the time, this was a monumental achievement in the field of membrane protein structural biology. We had sufficient resolution to visualize the transmembrane alpha helices in each of these hemichannels. So a hemichannel sits in the plasma membrane of one cell, and a second hemichannel in the membrane of another cell. And then these dock together through these extracellular domains that once docked form the complete channel pore. And so electron crystallography as a method had been demonstrated to resolve membrane proteins that have been crystallized in a highly ordered fashion at very high resolution. So work by Tamir Gonen had shown that aquaporin 0 2-dimensional crystals could diffract and give structural data beyond 2 angstroms. However, the same methods applied to these channels really were stymied in resolution and never really got beyond this kind of 6 to 7 angstrom resolution limit. And so when I started studying these channels in our lab at Portland State, we wanted to move into what had become kind of the go-to method, not only for soluble proteins, but also for membrane proteins, and that's single particle cryo-EM. And so we, in 2018, we published our first structure of these lens gap junctions using single particle methods. But we had the caveat still that we had removed these channels from the native lipid environment, and we solved the structure in the presence of an amphipole. And so moving forward, we really wanted to be able to study these channels in a more native-like environment, and we started to take advantage of these lipid nanodisc technologies that have become quite popular in the field. And the particular technology that we've had the most success with is the one illustrated here using these membrane scaffolding proteins, which are really just engineered versions of apolipoprotein. So they're an amphipathic alpha helix that under appropriate conditions can self-assemble in the presence of lipids and encapsulate them in this disc-like particle. And if you perform these reconstitution experiments in the presence of your favorite membrane protein, you can ostensibly reconstitute your membrane protein into this kind of solubilized lipid bilayer system. So that was our goal. And so to get started, like with what I tell my students and anyone interested in joining our lab, before you can drive the fighter jet, you have to become a biochemist. And so everything starts with biochemistry. I really consider this the most important part of any structural biology project. And for our lab, we really like to try to work with the most native protein possible. And so we isolate our protein from native source. We obtain eyes from model systems, either cow or sheep. For the particular work I'll talk about today, we've been working with sheep. And through a lot of kind of traditional biochemistry. We've optimized purification protocols to isolate native lens gap junctions. And so in the lens, there's two isoforms that are expressed, abundantly expressed. These are isoforms Connexin-46 and Connexin-50. We've shown that these two isoforms co-assemble in the lens into the same channels and form multiple types of heteromeric or what are known as heterotypic assemblies. Although the precise pattern of co-assembly and the variability of this kind of co-assembly is still something we're trying to understand. And so I should mention that these two students shown up here really led the way on the biochemistry for this project. In particular, I'll mention Kim Dolan, who was an undergraduate student in the lab at the time, and really kind of took on the challenge of then trying to take these purified proteins and reconstituting them into our lipid nanodisk system. And so we're certainly not the first people to work with these lipid nanodiscs, but the challenge here was that we have a membrane protein that needed to be coaxed into two of these nanodisk systems simultaneously. And as far as we're aware, this is at least the first reported success of trying to reconstitute such a complex system. And Kim really worked hard over at least a year of screening various different types of MSPs and many different types of lipid to protein ratios until we really optimized these conditions very well and got very clean biochemistry. And so this is a size exclusion chromatography trace showing our optimized reconstitution. We very cleanly resolve our reconstituted channels and nanodiscs away from the empty nanodiscs. And a really key tool to this optimization procedure is including negative stain EM. So this is a traditional way of preparing samples to be visualized on an electron microscope. A lot of labs are moving away from this kind of traditional method as part of their routine characterization, but we still maintain that this is kind of a necessary aspect of our preliminary characterizations and optimizations. And for the nanodisc reconstitution work, it was really informative because a lot of the contaminations you get in these reconstitutions are empty vesicles, which don't necessarily show up in these gel filtration traces. And so you may be finding that you have a lot of contamination from large vesicular lipids that would go unnoticed if you didn't include this type of analysis. And so once this was optimized, we're quite happy with the biochemistry, and so we're able to move on to imaging, and we prepared grids and put them in the microscope and started taking pictures. So I'll show you a representative of one of those micrographs shown here. And I'll highlight one of the particles. So we're looking at a side view of the particle. They kind of look like little butterfly-like structures. And for those of you who are familiar with CryoEM, you know that the major obstacle to this technique is the very poor signal to noise that you obtain from these micrographs. And so to overcome this, it's really just a numbers game. And so we take advantage of signal averaging and we collect very large image data sets in order to enhance the signal of these micrographs. And so the data I'm be telling you about today was collected on a Titan Krios using a Falcon 3 detector. We collected you know, a relatively modest data set. The goal of this project really wasn't to go and push any resolution boundaries. We just wanted to see what these channels looked like in a lipid environment. And so we collected a data set of around 2,000 micrographs. From that, we extracted about 1.2 million particles. I'll say particles in quotes because a lot of those initial particles are kind of not true particles. We very quickly clean this up to a data set of around 260,000 what I would call good particles that look nice in 2D class averages. And through a lot of processing and removal of imperfect particles, either through 2D classification and 3D classification, we ended up with a final data set of around 26,000 particles that went into the final structure that I'll be showing you today. And everything was processed in RelyOn, and the final processing was done in the most recent release of RelyOn in 3.1 beta. And so, you know, the first indicators of how the data is going to look comes from the two-dimensional projection averages of your data set. And for me, this is really the most exciting part of the image processing pipeline. This is where I hang over my students' shoulders and ask for constant updates, because you can really tell a lot about what your data is going to do by looking at these images. And so I'll show you some of the 2D classes that we got from this data set. So you can see some very nice high resolution features in these 2D particle classes. And the other aspect that we were happy to see is that we had nice angular coverage. So we had not only side views and some tilted views, but even some end-on views that are all helpful for achieving high resolution. And you know, just to give you an appreciation of the quality of these 2D classes, I'll zoom in on one of them and talk about some of the details. And so these two lobe-like densities that you see at the top and bottom are the 2 nanodiscs that we reconstituted into. So we're happy to see that they were the right dimensions for our nanodiscs. And if you measure the distance separating these 2 nanodiscs, we had, you can measure the same 3.5 nanometer spacing that is observed in micrographs of cell sections, indicating that we had captured, we hadn't severely at least disrupted the structure of these channels. You can see this nice lattice-like structure at the extracellular docking domains. And then if we go into these striations that you see crossing the nanodiscs, these are the transmembrane alpha helices. So we're visualizing secondary structure. And even beyond that, where we really start to get excited about these 2D classes is we're not just seeing rods. You can actually visualize the helical pitch in these alpha helices as they stretch across the lipid bilayer. And so just from these images alone, we really knew we were onto something kind of special early on in the image processing of this data set. And in fact, the morning I saw these, I was so excited I had to text my wife a picture. You know, so my wife Susie actually works in our lab, and she's on this project. She works with the gap junction, so she knows what she's looking at. And I thought her response was quite fitting for the title of talk. So she says, "Holy shit, 1 angstrom." If you know Susie, if you know my wife, she's not one to throw out swear words. So she was very excited. Now me, on the other hand, I'm a little bit looser with the English language, and so my response, I think, kind of exemplifies the excitement that we had in the lab at that moment. And that's because we knew that with data of this quality, we're onto something special. What I really didn't expect, though, was how far this data was going to go for us. And I really credit Jonathan Flores, a graduate student in the lab, who really did all he could to eke every bit of information out of this data set in order to obtain this 3-dimensional reconstruction, which he was finally able to push to a final resolution of 1.9 angstroms. And so in this inside here, I'm showing you a zoom where you can see all the high resolution details that we're able to see for the protein structure. You know, we see beautiful holes through all the aromatics, all the backbone density, and able to build in a really detailed and highly refined structural model for the protein. And for me, what's really exciting when you start to get to this resolution is all the other things that you see. And so these red dots that are speckled throughout the protein structure are ordered water molecules that we ended up resolving at this high resolution. And now, you know, being able to see these waters is an important aspect of structural biology. I teach biochemistry at Portland State, and you know, what I teach my students is water should really be treated as, or considered as, the 21st amino acid, because of the integral role that waters play in facilitating both the structure and function of membrane proteins. And so you can see all these beautiful solvent densities that we see resolved throughout the channel. And then, of course, the other really kind of striking thing that we could see in this reconstruction are these blue lipid densities that are kind of ordered like bouquets at each of the Connexin subunit interfaces, and have this kind of long range, apparently long range stabilization of the lipid environment. And so I'll come back to talk about the details and insights that we've gained from some of these new features. Before we get there, I want to talk a little bit about some of the protein structure and the several checkpoints that we do in the lab to kind of validate the structure and make sure that what we're looking at is actually meaningful. And so Johnny was able to take this map and build his atomic models. And so we get a representation like the one shown here. And so I'll remind you that the sample that we purified from the lens is a heterogeneous mixture of these two isoforms, Connexin-46 and 50. And Johnny was able to build models for both of these isoforms into the same map. And his ability to do that was only really possible because of the close structural similarity of these two isoforms. And so at the amino acid level, 46 and 50 share 80% sequence identity. And so I've highlighted some of these amino acids in white here where these two where the residues are identical between these two isoforms. And so you can see that in these cases, both models obviously fit well into the map. Both aromatics, long chain hydrophilic residues. I really like this example here where we can even resolve the whole through this 5-membered proline ring. However, then at these sites of dissimilarity, the fit isn't always quite as nice, and I've picked out some particular examples just to highlight the effect of this heterogeneity on our reconstruction. And so there's some examples that I think are kind of interesting. So for example, this leucine and isoleucine at these two positions here, you can see some evidence of both of these residues at these sites. In other cases, we have kind of a loss of signal or missing density at points at which the residues are non-identical. And so the reason for the quality of the map at these two sites is really, we believe, twofold. One is obviously a result of the heterogeneous mixture that we have by reconstituting both isoforms. However, it's complicated because a lot of these residues where these sites are different are also solvent exposed in hydrophilic amino acids that you'd expect to be flexible. And so for example, this position 9 we know as a highly dynamic site in the protein. We actually have a preprint coming out hopefully in the next couple of weeks through a collaboration with Doug and Bai, where we've been characterizing the structure and dynamic and functional role of this site. And so however, what we can say is that at the end of the day is that these two isoforms really do share a remarkable level of structural homology that is the only way that made it possible to reconstruct at such high resolution. Okay, so now I'll take the structure and I'll show you a top view down the channel pore. And so the pore of this channel is approximately 1.4 nanometers in diameter, and this is consistent with capturing the protein in an open state conformation. I'll point your attention to this alpha helical domain that folds into the channel vestibule shown here. So this is the N-terminus of the protein. We refer to it as the N-terminal helix domain. And we had shown previously in our amphipole work that this domain folds kind of a classical amphipathic alpha helix. And so there's a hydrophilic face to this domain that packs against the lumen of the channel. And this positions these hydrophilic and charged amino acids towards the vestibule, where they can play a role in substrate selectivity. And this domain is thought to function as a gate. So there's a hinge here connecting it to the first transmembrane domain. And the idea is that this NTH domain can flip open and closed. And many of these charged amino acids have been implicated in the voltage gating of these proteins and in substrate selectivity. And so the architecture that we captured here seemed very fitting with the function of this domain of the protein. And when we first resolved these channels, what was surprising to us was comparison to the previously described crystal structure of a related channel, Connexin-46. And the most striking difference between our structure and the previous X-ray structure really came down to this domain. And so I'm showing the motif from Connexin-46 and 50 here in red and blue, and then Connexin-26 here in gray. And what was striking is many of these conserved hydrophobic amino acids in Connexin-26 were not pointed at the at the interface, but rather into the solvent, which didn't indicate that it was in a very stable or happy conformation. And we performed MD simulations on these channels, and what we observed very rapidly is that the Connexin-26 conformation was really not stable by MD simulation as compared to the conformational state captured by Cryo-EM. And so what we believe, we still don't know what the physiological role may be for the state that was captured by crystallography, but what we've been saying here is that at least for 46 and 50, using Cryo-EM, we've been able to capture these channels in a stabilized open state conformation. And that same conformation was captured in the presence of our lipid environment. And so we're happy to see that. Okay, so we're happy with the protein, but at the end of the day, the conformation of the protein that we captured in the lipid environment, although going to much higher resolution and allowed us to refine the structure quite a bit, really wasn't strikingly different from what we saw in Amphipool. And what was most striking, of course, then was all these additional features. And so we'll talk a bit about those. And so the first thing I'll talk to you about is all these red water molecules that are decorated through the channel, and give you at least a brief idea of some of the structural and functional roles that we think that these water molecules are playing. And so one of the most interesting sites that we see a lot of decoration of ordered waters is lining the permeation pathway of the channel. And so many of these hydrophilic and charged amino acids that line the pore of these channels, the specific conformational state of these amino acids is sort of cemented or held in place through this tight network of water molecules that that kind of buttress the conformational state of the channel. And so we believe that these water molecules are playing an important role in facilitating the permeation mechanism. We're interested now in trying to simulate the permeation of various types of molecules going through these channels to understand better how these water molecules are playing a role. We have done quite a bit of simulation with different ions, and many of these charged residues appear to play a transient role in interacting with ions as they permeate the channels, and these water molecules likewise play an important role in facilitating some of these transient binding sites. And so we think they do play an important role in the transport mechanism of these channels. So the other area where I'll highlight is at the docking interface of the gap junction. So I'll remind you that we have a one Hemi channel here and another one here, and they communicate through these extracellular domains. And so we'll zoom in on these extracellular docking sites. And at the interface of two Connexin subunits, there's this cluster of buried water molecules that are highly integrated into the hydrogen bond network that facilitates this kind of molecular recognition that takes place between these two Hemi channels. And so I mentioned that there's 21 different Connexin isoforms in the body, and they can co-assemble in different ways, but not all isoforms can co-assemble with one another. There's there's some connexins that can co-assemble and some that can't. And so there's rules to this molecular recognition that your body obeys. And this is thought to play an important role in deciding which tissues communicate with who. And so understanding this type of molecular recognition and the principles guiding this molecular recognition is really important to try and understand how our bodies kind of coordinate long-range communication through the gap junctions and between, not only between cells, but between different tissue types. And so it had been proposed that specific hydrogen bond patterns between different isoforms that are compatible would play a role in deciding these rules between which isoforms communicate with who. However, what we see now is that there's a huge network of these water molecules that we think also play an important role in guiding this kind of connexin specificity. And so we're just now starting to try to understand the precise role that these waters might play in dictating which connexins can talk to who. And so at the end of the day, we ended up resolving almost 400 water molecules throughout the structure. Almost all of them could be interpreted as playing some sort of important functional or architectural role. Right, so then the other aspect of the channels that were really quite exciting. So these blue structures, the lipid molecules. And so I'll highlight two of the kind of striking, at least what I found as striking features of these lipid densities that we ended up modeling. So we have two lipid nanodiscs, and each nanodisc is a bilayer. And what was really surprising is really the only lipid density that we saw corresponded to the lipids that were specifically at the extracellular leaflet of these two membranes. Right, we didn't really see any lipid density in the intracellular leaflet. So it indicated that this interaction with the lipid environment was at least leaflet specific for these channels. And then the other striking feature is kind of the long-range stabilizing effect that these channels had on the lipid environment. And so we not only visualized the annular lipids, the lipids that directly interact with the protein interface, but we see second, third, and even fourth order stabilization to the lipid environment. And this was really kind of surprising. I'm yet to be aware of another example, at least from a structural biology point of view, where this sort of stabilization had been captured before. And so we're very intrigued as to, what was influencing this and what might be the kind of biological impact of such level of stabilization. However, when we first started showing these structures and talking about these results to people, the first critique that we always got was with the lipid that we chose to do our reconstitution studies with. And so we chose to do our reconstitution studies with this model lipid DMPC. And so DMPC is very nice. It's a phosphatidylcholine. The lens is rich in phosphatidylcholines. However, it's got some unique features that that for lipid biophysicists raise some eyebrows. So the first feature is that they're completely saturated and they have relatively short ACL chains. And so both of these components lead to a relatively high melting temperature for these lipids. So they have a melting temperature that's just below room temperature. And we did these reconstitution studies at around room temperature. And so there was concern that perhaps what we were seeing was just biophysical effect of the particular lipid. However, my first argument to that was that if we were really just seeing a phase transition of the lipid itself, we should have seen the same effect in both leaflets. And the fact that we were seeing the effect just at the extracellular leaflet really led me to believe that what we were seeing was a protein-specific influence on the lipid environment. Nevertheless, we wanted to understand this system a little bit better. And so we called in a very talented student in my lab, Bassam Haddad, who has really spearheaded a lot of the molecular dynamics simulation work in my lab. And so we asked him to simulate these channels now in the presence of DMPC, but at a more at a much higher temperature than what we could do with our reconstitution studies. So we did all of our MD at 37 degrees Celsius, and we asked, and we didn't want to have any kind of um, um, we wanted this to be kind of a blind study, and so we asked Bassam to when he built his system, to use a completely random placement of lipids. He didn't use the coordinates of our lipid system to build his MD simulation. And what was really striking is that following equilibration of the system by MD, we ended up resolving. So you can generate time average densities from your MD simulation, and we overlay those densities with our Cryo-EM map. And we saw almost exactly the same type of pattern of stabilization to the extracellular lipid leaflets, even simulated at 37C. I really thought this was striking. So we see this kind of same hexagonal packing pattern of the lipid densities that we see by Cryo-EM. And even to, like, we try to contour these things at roughly the same level, and you see almost the same extent of stabilization that we saw by Cryo-EM. So this kind of cooperation between these two methods was really quite striking. And so we went on to analyze the MD data in a little bit more quantitative way. And so one of the parameters that we can extract from the MD data is something known as a lipid order parameter. This is something that's stolen from the NMR community, but it can serve as a reporter for the phase state of the lipid system that you're simulating. And so we've tried to color these by the lipid order parameter. So blue would be consistent with an order parameter of a lipid that's in the gel phase, and white would be consistent with something that's in the liquid phase. And so in the bulk lipid system, we see what we would expect of the lipids behaving like they're in the liquid phase. But then this kind of, again, striking specificity for the extracellular leaflet of ordering these lipids towards the gel phase, and not, again, just not only at the annular lipid interface, but this kind of long range stretching out to around beyond 20 angstroms of stabilization to this domain of the lipid bilayer. So we can look in a little more detail as well with the MD simulation work. And so we're zoomed in on part of the structure here. This is the MD lipid density. You can see kind of having that same pattern that we saw by Cryo-EM. And what Bassam did here is to fit several of the symmetry-related lipids into the same density here. And when I start the simulation, you'll notice two things. One, you'll notice this kind of ordering of the ACL chains that looks like this kind of gel phase, kind of rod-like conformational state. And in contrast to that ordering that we see in the ACL chains is this highly dynamic behavior of the lipid head group and glycerol backbone. And so this was interesting and actually informative, because one of the features of the Cryo-EM map that I really haven't highlighted here yet is that in the ensemble Cryo-EM data, we didn't observe any lipid head groups. So all these lipids that we modeled here are just the ACL chains. And we didn't see any head group density, and the MD simulation is consistent with why we didn't see that. And that's because this part of the lipid environment remains highly dynamic. However, within this ensemble, we did see some kind of favored states of the phosphatidylcholine head group. And so Johnny decided to go back to his Cryo-EM data, the ensemble Cryo-EM data, and really try to interrogate that heterogeneity using the really beautiful capability of RelyOn and doing these kind of 3D classification methods. And so he performed several different approaches to 3D classification. The approach that worked best is displayed here. When he separates out his data set using this approach, you'll see that most of the data actually goes in this class here, 80% of the data. However, in this data set was just like our ensemble Cryo-EM data set, we don't see any lipid head group densities. However, if we looked in detail at some of these other smaller classes, we could start to see some density that was consistent with the head group of some of the annular lipids. And we were able to classify out some different conformational and configurational states for some of those lipids. And so I'll show a zoom up view of that. And so I've overlaid the 3 different classes that we're able to pull out of the data set here in different colors. And so what was striking about this was that all 3 of these classes actually share overlapping structural features. And so what I mean by that is if we take kind of a cross-sectional view of the lipid, you'll see that, and we've numbered each of these lipid densities just for kind of indexing purposes. And you'll see that this first class here occupies the 1,5 configuration. The blue density, the blue lipid occupies the 1,2 configuration, so they have an overlapping placement of the ACL chain at position 1. And likewise, the orange lipid here has an overlapping configuration of one of its ACL chains with the blue lipid. And then if we look in more detail, class 2 and class 3 lipids actually share similar placements of their choline head group. Now, what was again interesting when we look at the kind of the details of these conformational states is these kind of pseudo-stable conformations are all facilitated by non-specific interactions with the protein. And so this choline head group is supported by backbone carbonyl hydrogen bonding, as well as interactions with some of these stabilized water molecules. The class 1 here, we don't visualize the choline head group. We do see the phosphoglycerol backbone. The backbone's interacting with water molecules, and the phosphate group seems to be pinched between these 2 positively charged amino acids. Right, so these kind of alternative configurations that we're able to visualize, again, all fitting with the idea that these interactions with the protein are relatively non-specific and heterogeneous. And so this was interesting. So we kind of did this back and forth between the Cryo-EM and MD work. And so we then took this result and went back and reanalyzed the MD data to see if some of these prominent configurational states that we observed by Cryo-EM were also kind of the more stable states that were observed in our MD simulation. And so the two of the representative configurational states that were resolved by Cryo-EM, what I'll call the 1,2 state and the 1,5 state, showed up consistently as being some of the more stable configurations that we observed by MD. And so I'll play the simulation for you here. This is really similar to the movie that I already showed, where you'll see that the ACL chains kind of sit within this kind of defined density, while you can see in the background the phosphatidyl head group is just kind of flailing around in the background. But they stay in this kind of configuration throughout the simulation, indicating some sort of relative stability for this configurational state. However, if you go look at other examples throughout the channel at different symmetry locations, we can look at those exact same configurations and see that they don't always just sit in that same spot. Right, so I'll have you pay attention to this particular inset. So we're going to start what I call the 1,5 configuration. And as the movie starts, you'll see that it begins to transition. This ACL chain starts to snake its way over to this, the 2 position, and then the other ACL chain follows, and it kind of stays there for a little while. And then before the end of the simulation, it's going to transition back to the 1,5 configuration. And so, there's several other examples of this kind of behavior where, although the lipids seem to be relatively stabilized by interactions with the protein that kind of favor this gel-like phase of the ACL chain, they're not locked into rigid positions. They can transition between different configurational states, and the time scale of these MD simulations is relatively short. So we simulated only for around 100 nanoseconds. And so these transitions are happening relatively rapidly. And so this is still a very dynamic environment. And what we concluded is that Cryo-EM was able to capture, I guess, representative poses of these lipids from an ensemble dynamic of different configurational states. And so I thought this was very cool, and I think kind of moving forward, I think it speaks to the capability of Cryo-EM, not only to resolve kind of dynamic ligand interactions like these physiological ones shown here, but also from the point of view of structure-based drug design efforts, where you can envision perhaps even doing what crystallographers have been doing for decades now, is even mixing your sample with an ensemble of different ligands and then pulling those out using different classification methods to identify which ligands or poses are bound to your structure. And so before wrapping up, I'll just talk a little bit about some kind of broader insights that I think that we're starting to appreciate or think about with this system. And so I'll go back to this image that I showed previously of these large arrays of connexin channels or gap junctional plaques. And if you consider this context as the kind of native and context of how gap junctions exist in our body, and then the influence that we observed in the lipid environment, we can start to stitch together a model that might look something like this. And so I've placed this is just a hand-placed model, but I've roughly placed the structures at a spacing that's been observed in kind of the lattice spacing that's been observed inside of cells. And so you can see that the extent of stabilization to the extracellular leaflet would really kind of result in an entire stabilization of the plasma membrane along that leaflet in these kind of electrical junctions or gap junctional plaques. And so what is the physiological role of this? We really don't have a firm understanding yet. I think it could be many-fold. You can imagine it being an important aspect of maintaining an electrochemical seal at these interfaces. One of the things that we're interested in, and we're starting to discuss with some lipid folks, is how this may play a role in lipid sorting and for example, in lipid raft formation. The gap junctions are known to be enriched in lipid rafts, and the types of lipids that are abundant in lipid rafts, such as sphingomyelin, have a relatively higher melting points and degree of saturation, similar to what we see with DMPC. And another kind of intriguing postulation is that this kind of hexagonal packing that we see in the lipid environment may serve as a sort of templating feature for organizing the hexagonal type of organization that we see in these gap junction arrays. And so it's exciting to think about, but challenging to kind of come up with experimental ways of validating some of these predictions. But we're starting to work with some really clever lipid biophysicists and biochemists to try to understand some of these things. And then, of course, having this high resolution system, we're poised to really make some detailed understandings of the function of these channels. There's still a lot we don't understand. These channels are, as I mentioned, they're gated in various ways, and we're just starting to make headway into understanding how that happens. And then trying to understand the effect of the lipid environment, both from a physiological and potentially pathological point of view. And then another desperate need in this field is for the development of more selective inhibitors. And so we think we have a nice platform to contribute to that, to just start with understanding the pharmacology of these channels, and then ideally contribute to the development of more selective drugs to both study and potentially treat diseases associated with these channels. And so I'll end here with just giving a quick acknowledgement. I try to do that along the way. Johnny really spearheaded the reconstruction and modeling of these channels. The Sam did the MD work on on this project. She's not pictured here anymore, but Kim Dolan, the talented undergrad in our lab who really worked out the biochemistry and also contributed to the early days of processing on this, but she ended up going off to grad school and is now at a program in Berkeley. Jeanette Myers is a former PhD student in my lab who helped with some of the image processing and getting Johnny off the ground early on. And a special shout out to my colleague, Dan Zuckerman, who's been a great collaborator over the years. And Dan is a biophysicist and theoretician and has really helped to train Bassam and oversee a lot of the MD work that goes on in the lab. And his postdoc, Jeremy Copperman, who really helped design some of the analysis that we did. And then lastly, a great friend, Craig Yoshioka, and colleague at the Pacific Northwest CryoEM Center. Craig is a guru of image processing and has been a great resource for our lab to really learn all the tricks of eking out the most we can from our data set. I think that's it, and I apologize if I missed anybody, but with that, I'll turn it back over for any questions we might have. Sorry, I think I'm looking at my clock now. I realize I'm going a little bit long.
No worries. That was a great talk. And if you don't mind staying for a couple minutes after, if we have more questions, that might be great.
Absolutely, I'm happy to.
So if you have any questions, you can send them to Wolfram or I in the chat. I guess one question I had is, so if you're doing an in vitro purification, did you see any evidence for post-translational modifications that might be a factor with either the gap junction formation or the oligomerization?
Yeah, so we characterize these quite a bit by mass spec to understand the post-translational modifications. And so what I didn't mention is there's significant portions of these proteins that we don't visualize in the reconstructions, and these correspond to cytosolic regulatory domains. So there's a significant C-terminal tail and an intracellular loop on these channels that are highly modified by post-translational modifications, mostly phosphorylation, but some other what I think are probably more related to aging of these proteins. It's not clear that these modifications play a significant role in controlling the heteromeric assembly of these channels, although I don't know that that's really been investigated very thoroughly. These do have an N-terminal acetylation that happens co-translationally. They also lose their very first amino acid during processing. And so that's something that we incorporate for our MD simulation work to kind of capture the effects of that neutralization of the N-terminus, because that would have a big effect on the energy barrier to permeate ions or substrates. But beyond that, you know, there's no glycosylation of these channels to, you know, the idea being that if you had glycosylation, you wouldn't be able to form a complete junction. So this is in contrast to the Connexins, the related channels. They typically are glycosylated in their extracellular loops, which is thought to prevent them from forming junctions.
So
yes.
Wolfram, you said.
So we have a question from Alex Payne, and Alex, you may unmute yourself and ask the question yourself.
Sure. This is thrilling work. It's so cool to see the combination of Cryo-EM and molecular dynamics to understand how this is working. But I was curious earlier in the pipeline, so the isoforms, are they randomly sorted through the whole oligomer, and were you able to use different sort of symmetry approaches to getting to higher resolution, or are they random and so it's hard to sort through that?
Yeah, great question. So the truest answer is we don't know. My best understanding is that it's if it's not random, there's at least many different ways of co-assembling that are present in the lens. So we know that there's both, so in this field we refer to heteromeric and heterotypic assembly. So heteromeric would mean you have more than one isoform within a hemichannel, and heterotypic would mean that you have different isoforms between the two hemichannels. And so using different types of, we can do pull downs and different types of biochemistry. We've done cross-linking, we've done native mass spec with our collaborator, Carol Robinson out at Oxford. And we detect heteromeric, we detect heterotypic, we detect at least more than one type of heteromeric pattern. However, the work that we did with Carol's lab really wasn't, so they're of course leaders in this approach of native mass spec, where you try to determine the mass of your protein without dissociating the native complex. And they've worked out methods for doing this with membrane proteins, where they can first dissociate the lipids so that they don't contribute to the noise, and then try to study the membrane channel. However, with our particular system, it was just the amount of energy that they had to ionize to knock off the lipids or the detergent was too strong, and we ended up dissociating most of the protein. And so we never had really kind of publishable results, but the data did show that we had at least a couple of different types of heteromeric assembly going on. And so from an image processing point of view, we've tried to process these things in using various different types of symmetry operators, as well as C1 symmetry. We've tried to do kind of this symmetry expansion and signal subtraction approaches to try to pull apart just one subunit and do three-dimensional classification on the isolated subunits. At the end of the day, none of these approaches have really worked for us yet, even with this high-resolution data set. And it really comes down to the fact that the points where these proteins are different, even if they're different chemically, those amino acids could be similar sterically. So, like a threonine and an isoleucine have different chemistries, but their densities look very similar, right? And then the other difficult part is where they are different tends to be at sites where the amino acids are flexible. And so we just don't get good density there in the first place to kind of tease apart this heterogeneity. And so moving forward, we really need to develop a fragment antibody system that we can label the two different isoforms and use the approaches that have been done, for example, with NMDA receptors and other channels to elucidate this type of heterogeneity.
Wow, thank you.
Michael Oldman, can you ask your question, please?
Sure. Great work and great paper. I have a question about just a simple thing about the methods. You did a subsequent round of auto picking using your initial map, and you made projections and oriented that in different fashions. Did you do anything special about which program did you use to make the projections, and which did you do anything about filtering the projections or injecting them with noise, for example, or just use them as they came?
Yeah, so this was all done internally with RelyOn. So
okay.
we kind of bootstrapped our way in using the Lipasian picker. We got initial data set and reconstruction, which ends up being filtered to low resolution, and we use that as then a template to go and do template-based particle picking. Yeah, and so yeah, so the model's generated from the data itself and filtered to low resolution. Does that help?
Well, I mean, how'd you actually make the back projections?
This is all done internally in RelyOn.
Okay.
Yeah.
Isaac Garcia, you may ask your question, please.
Hi, Steve. I'm Isaac Garcia from Universidad Valparaíso, Chile.
Hi.
Great work. I'm wondering to know if you are these water and lipids that are rearranged in gap junction channel are also observed in hemi channels, or is necessary to be present in hemichannels to dock the another one, or just are ordered
Yeah,
in the
so
gap junction?
yeah, so the waters that we see at the docking interface.
Yeah.
That's a great question. You know, so we don't have a structure yet of the hemichannel. We'd like to have one. And so if
Me
those
too.
waters are already ordered at the interface, would be yeah, that would be super cool to find out. And that would really hint at the idea that they play a role in this kind of molecular recognition mechanism. We also, you know, so other labs have simulated the hemichannel by just breaking the gap junction in half and simulating it that way. We haven't done that. I'm really not comfortable with that approach because, you know, I don't know that just breaking the gap junction in half is a good representation of what the free hemichannel looks like. And so, but certainly that would be a mechanistic question I'd like to understand, and something that we'd certainly be interested in trying to visualize by CryoEM.
Okay, can I make another one?
Sure, go ahead.
Question? Okay, thank you. And also, do you find any evidence that the N-terminal is something, the flexibility is affected by this water and lipids?
Yeah.
In terms of the function of the channel, you know, the N-terminal is a sort of plug that goes inside the pore and modulate the permeability of the channel.
Yeah. You know, so this is another aspect that we're very interested in. So if I can pull real quick, right? So you're still seeing my screen? Yeah. So the N-terminus is kind of sitting right here inside of the channel, and this would be kind of most directly adjacent to the intracellular leaflet where we didn't see any lipid densities, right? And so this is, you know, one of the things that this structure is kind of indicating is that these channels interact differently with the extracellular leaflet and the intracellular leaflet. And so moving forward, we'd really like to understand, you know, what, if any, kind of specific interactions the channels might be having with a different set of lipids, for example, in the intracellular leaflet. And does that leaflet have particular influence on the gating domain? There's been reconstitution studies of not with these isoforms, but other related isoforms in different lipid environments, which have been shown to affect channel function. And so there's certainly an influence of the lipid environment on the permeability and gating of these channels. And that's something we'd really like to explore and understand better, for sure.
Okay, thank you.
Alex Payne has a follow-up question.
Yeah, about the lipid ordering. Is there knowledge of, like, using chemical probes to get more in vivo lipid order parameter data for how the connexins are stabilizing the plasma membrane?
Yes, there are, and this is work I've just become familiar with since we started. You know, this, you know, the results kind of your research starts to get you interested in other people's research, and so there are probes for this, and that's something we certainly want to explore and see if we can use to understand in a little bit more detail. Yeah, in both reconstituted and ideally in kind of a native cellular system, what level of ordering are the gap junctions doing to the lipid environment? And so that's something that's a direction we certainly want to explore. They have, there's particular fluorophores, for example, that their fluorescence is modulated by the phase state of the lipid environment. So they can be used as indicators for the phase changes in the lipid environment.
Yeah, that's very cool.
Yeah.
I'll be excited to see the results of that.
Cool.
And we have a question from Sumit Bandekar. So.
Hi, Steve. Great talk. So I guess I had a question as to like biologically, like what are the prerequisites for a productive connection channel formation? Are there prior cell-cell adhesion processes that need to occur to ensure productive channel formation, and so on?
Yeah. Yeah. No. Good question. You know, so I can say from like a non-native setting, the only thing you need is these cells expressed at the plasma membrane. So this comes from, for example, electrophysiology studies,
Very cool.
right? So if you have gap junction deficient cells, so the way you do electrophysiology on these studies is quite unique as well, right? So you end up patching two cells and either bringing them together or isolating two cells that have already formed gap junctions, and you pass electrical current between them. And if you have gap junction deficient cells, they don't form these adhesions. But if you express them on the surface, then that's all that's necessary and sufficient to form interconnected cells.
Thanks. Well, if there are no more questions, then I guess we're done for the day. And Steve, thank you again for a great presentation.
Yeah, thanks for the invitation. It's been a lot of fun.
Well, one day we have to figure out a good way to do clapping over Zoom. Like, you know, it doesn't seem to work. Maybe there's the Zoom wave. I don't know. But Alex has a nifty clap emoticon. So.
All right, wonderful. Well, thank you all for joining. I really appreciate it, and hope to see you all in the real world again someday.
Likewise.
All right. Take care, Peter.
Take care. Thank you.
Bye.